Hello and welcome back to our series of lectures about sensory systems. In this video, we will be talking about audition, which is our sense of hearing, and equilibrium, which is our sense of balance. Okay. All right, so before we talk about how we detect sound, I first want to talk about the nature of sound, right? So in order to understand how we detect sound, we have to understand what sound is exactly. And that's what's diagrammed in this figure right here. Basically, what sound is it's waves of alternating peaks of compressed and non-compressed air, right? So you have this like sort of compressed area of air here, non-compressed, compressed, non-compressed. And that's basically the nature of sound waves. And that's what's coming to your ear. And that is what we are going to detect with our ears. So let's take a closer look at the ear. I know this is kind of a messy looking figure, but oh well, it is what it is. So the ear has three main parts to it. We have the external ear here, the middle ear, and the inner ear. The external ear consists of, you know, this part out here that you usually see in the ear canal. It is separated, sorry, it's separated from the middle ear by this membrane, the tympanic membrane, which you might know as your eardrum. And then the middle ear is separated from the inner ear by a membrane in here called the round window. And there's actually, sorry, okay, so there's the oval window up here and the round window down here. So both of those connect the middle ear to the inner ear. The external ear and the middle ear are air-filled, while the inner ear is fluid-filled. The inner ear actually consists of, or it contains both the cochlea, which is our main organ for hearing, and then it also has the vestibular apparatus, which we use for equilibrium. Okay. All right, so let's take a look at how we detect sound in our ears. So we start with sound waves coming in through our ear canal. Those sound waves vibrate the tympanic membrane or the eardrum, okay? There we go, okay. The tympanic membrane is connected to these three bones right here, the malleus, the incus, and the stapes. Don't worry about the names, but just, you know, remember that there are these three bones here. They're the smallest bones in your body. So the vibrations of the tympanic membrane are carried through the middle ear by these three bones. And as they carry that signal through, they amplify the vibration, okay? Then that vibrates the oval window. So the stapes here is connected to the oval window. That's going to vibrate the oval window. When the oval window vibrates, that vibrates this fluid here in the cochlea, okay? All right, so now let's take a look at what happens within the cochlea. And to do this, I want you to imagine that we are taking a cross section like this through the cochlea, okay? So we're going like that. And then we're going to turn it 90 degrees towards us, okay? So we're gonna go like that and then turn it, okay? Oh, sorry, I forgot to put those up, okay. So if we do that, oh, sorry, going back here. So if we do that and we look at sort of the bottom of this blue part right here, so there's this duct here called the cochlear duct. If we look at the bottom of the cochlear duct, this is what we're gonna see. We have basically a membrane at the bottom that's kind of basically connected to the bottom of the cochlear duct, okay? Embedded within that membrane are these cells called hair cells, and those are the ones that are going to be doing the detecting of the sound. Lying on top of those hair cells is this membrane right here called the tectorial membrane. Okay, so this is like extreme cartoon form, and then this is a less cartoony form, showing you the same thing, that we have these membranes here with the hair cells embedded in the basilar membrane, this membrane down here. Okay, so what happens, remember that we had, sorry, going back, remember that we had all these vibrations traveling through the ear. That's going to cause vibration of the fluid in the cochlear duct. When we get that vibration in the cochlear duct, what happens is these two membranes, the tectorial membrane and the basilar membrane, vibrate in response to that fluid movement, okay? And they vibrate in relation to each other, okay? When they do that, they activate these hair cells, okay? So the hair cells, like the tips of the hair cells, are in contact with the tectorial membrane. So when these two vibrate, then we're going to kind of basically do this to the hair cells, okay? The next figure shows this in more detail, okay? So let's take a closer look at hair cells. This is what a hair cell looks like in cartoon form, of course. The hair cells have these stiff cilia at the top of them called stereocilia. And those stereocilia are linked to each other by these protein bridges called tip links, okay? And so when a hair cell is not activated, that's what's shown in this panel. We have, sorry, I forgot to mention, these tip links are connected to mechanically gated ion channels, okay? So when the hair cell is not activated, when it's at rest, some of these mechanically gated ion channels are going to be open, and we're going to get some neurotransmitter release from the hair cell, okay? Now remember that we have our tectorial membrane here lying on top of these stereocilia, right? And when we have vibration in the cochlea, that's going to vibrate the tectorial membrane, and it's going to move these stereocilia, okay, like this. So this next panel shows you that if we move in one direction, that's going to cause more of these mechanically gated ion channels to open. We all hopefully know by now what happens when ion channels open, right? Ion channels open, we get ion movement across the membrane. That's going to lead to a change in membrane potential. In this case, that's a graded potential that travels down and is interpreted and leads to more neurotransmitter release, right? So we have a larger signal here than we had over here. So we get more neurotransmitter release here. And then in the cell that is downstream of that, we get an increase in our number and our frequency of action potentials, okay? Okay, so that's what happened when we moved the hair cells this way. Let's take a look at what happens when we move the hair cells the other way. When we move the hair cells the other way, those channels close. And as a result, we don't get that ion movement across the membrane. Our neurotransmitter release decreases, and our number of action potentials decreases. Okay, so that's how we send that signal.